Hi, I'm Carimetelo, an eighth-semester student of the bachelor degree in biology pharmaceutical chemist and the Faculty of Chemical Biological Sciences, belonging to the Autonomous University of Campeche, and will talk to you about CRISPR-Cas technology. For years, human beings has made many attempts to modify nature, including plants, animals, and microorganisms, in order to satisfy his economical, medical, environmental, and health needs. During the last year, genetic engineering technologies have had enormous growth in the field of the genome editing in order to generate new species with certain desirable characteristics. What does genome editing is? It's a tool that manipulates and modifies a DNA sequence. Modification including insertion, deletion, or replacement of base pairs. Why to edit genome? Genome editing can be really helpful in several scientific, medical, or industrial fields. For example, it can help to study genes, correct hereditary diseases, create new treatments for lethal diseases, develop better crops, and produce healthier food. How does genome editing works? It works by using certain enzymes which can cut a very specific part of the genome. After cutting the DNA in the specific place, the technique will take advantage of the natural cell machinery to rip the cut. This repairing process induces the change where the DNA was cut by adding, removing, or replacing nucleotides. Methods used for genome editing. The most common methods for genome editing are zinc fingers, TALEN, transcription activator like effector nucleases, and CRISPR-associated to Cas. What CRISPR is? CRISPR stands for Cluster Regularly Interspaced Short Palindromic Repeats. They are elements that bacteria use as an immunity mechanism that gives them protection against viruses. They are short sequences that derive from previous viral infection and have been incorporated into the bacterial genome. When the bacterium is attacked again, CRISPR is transcribed into RNA and made to a Cas protein. RNA Cas thin and viral DNA sequence, complementary to its own and Cas, cuts the viral DNA and inactivates it, stopping the ongoing viral infection. What is the Cas protein? It is a group of endonuclease enzymes that acts as a molecular scissor and cut DNA fragments in a very precise way. The most famous is the Cas9 protein. How does the CRISPR-Cas technique work? This bacterial immunity mechanism has been used since 2012 as a genome editing technique. The process is 1. We need to know the exact DNA sequence of the gene that we want to edit. 2. Then we have to design a RNA fragment complementary to this gene. 3. This RNA will act as guide for the Cas9 to find the DNA of the gene of interest. 4. The complex Cas9 RNA will find the gene, and Cas9 will cut the DNA fragment. 5. After cutting, the cell machinery will try to fix it. 6. This repairing process is prone to mistakes and may cause mutation. 7. CRISPR not only can cut DNA, but it can also add other DNA fragments to correct mistakes or edit the gene in a very desirable way. CRISPR is a genetic technique which is very simple, cheap, and efficient. However, there are ethical controversies side this manipulation, may have biological risks or even be used in work to create species that way represent the danger or take advantage over the rest. Application. In agriculture, for example, Materia Biotechnology has used in China CRISPR to produce rice by deleting a DAP1 gene. The result is an improvement in the yield of the crop. In the United States, CRISPR has been used to prevent mushrooms from burning by silencing the polynephrol oxidase genes. The product is waiting for approval of the FDA to reach the market. In medicine, for example, in 2016 in China was treated the first person against cancer using CRISPR by silencing the PD-L1 gene, which is involved in the immune response against cancer. In conclusion, CRISPR is a genome editing technique that represents an easier and simpler way to create new species or cells with a desirable purpose that can benefit humans in several ways. In my point of view, it's a good technique that might help in the future to treat very lethal diseases like cancer. I will give people a new chance to recover and save their life. Thank you.